Welcome to today's presentation on Chapter 3. Today will be the first of two discussions related to Chapter 3. Today we'll be taking a look at 3.1, the cell membrane, 3.2, the cytoplasm and cellular organelles, and finally the nucleus and DNA replication. Remember that the cell is the smallest, most fundamental living thing in your body. Your body is composed of trillions of cells. And if we go back and just briefly remind ourselves, back in Chapter 1, we saw the structural relationship of the body. And this is a really important organizational figure for you to really understand. That is, let's go through this backwards. So as an organism, as a human being, right, you are made up of organ systems. The other way of saying organ systems would be body systems. These are like the urinary system, the respiratory system, the skeletal system, the muscular system. Those systems, each of those 11 body systems, is itself composed of organs. In this example, the urinary system is made up of the bladder, as well as the kidneys that aren't shown here. And then what makes up organs? Well, organs are made up of tissues. Tissues, and there's 4 different types of tissues, and we'll be studying tissues a lot in Lab 2 as well as in Chapter 4, but there are 4 kinds of tissues. There are muscular tissues, there are nervous tissues, there are connective tissues, and there are epithelial tissues. These tissues are composed of cells. So cells combine to make tissues, and that's where we're going to be today. We're going to be talking about cells, and then the cells themselves are made up of organelles. Okay, organelles are the specialized little factories, the parts of cells, like the cell membrane, like the nucleus, like the endoplasmic reticulum, both smooth and rough, like the mitochondrion, the lysosome, the peroxisome, the centrioles. And today we're going to be focusing on those organelles and how they make the cell. Before that, the organelles themselves are made up of molecules, things like water shown here, but also other molecules. Look on your food label and you know that when you eat, you are eating proteins, you are eating carbohydrates, you are eating fats and lipids. Those are also molecules that are the building blocks of your organelles and your cells. And then most fundamental is that those molecules themselves are made up of atoms. Now, we are not going to be focusing on Chapter 2, which covers the chemical level of organization. However, you will see a little bit of information in Lab A, the asynchronous lab that you're all going to be working on over the next 10 or 12 days. That lab does cover some basic ideas from chemistry, a little bit about atoms, a little bit about pH. So you will see some of this in Lab A, and I will highlight the most important components of Lab A as all of you finish up that lab and before we take our first exam. So let's skip over again back to Chapter 3, and let's look at our first organelle, and that is the cell membrane. Okay, the cell membrane. Recall my suggestion to do a split screen so you can be watching this presentation on your computer, on your screen, and you could have your own copy of the Blossom opened up on the other side of the screen, taking notes, making highlights for your own study materials. Also, or you could have, for example, this lecture playing on your phone while you interact with your computer to make comments and make some highlights. So that's definitely my highest suggestion, is that you are following along in your own copy of the Blossom and making notations, making comments, making notes, and then you can go back and replay this video as often as you need to. Also recall that the expectation the assumption is that you have already glanced at this chapter. You have already read through it to the best of your abilities, and that as we come together to discuss it, that this is a chance for some of those main ideas to fall into place. Now, if instead you're listening to this presentation before you read it, that is another way of doing it. I don't know that it's the most ideal, but if you are listening to this for the first time, you're going to obviously be picking up on the main ideas. Depending upon the biology in your background, if you've had a significant amount of biology, either in the high school or college level, much of what I'm going to say today is going to be a review. For some of you, however, it's been a while. Or maybe you didn't take biology in high school. Maybe that just did not happen for you, or it's been so long that you really can't say that you remember much of what you learned. If that is you, that's fine. This book is beautiful in that it is designed to start you from the very beginning and build up your knowledge. Make sure that as you scroll through the Blossom that you are pausing to watch the videos. Make sure you are pausing to reflect and answer the questions along the way. Remember, you're answering the questions in the Blossom is a very vital and important part of your participation grade. And I don't want you to do it because you're getting participation points. I want you to do these questions because doing them is going to help confirm your understanding. It's going to allow you to bring questions to supplemental instruction and to lab, and it's really going to help you clarify and categorize the material that you're learning here. Recall that as chapters start out, we have the chapter objectives. This is for the entire chapter. For today, we're primarily going to be talking about the cell membrane. We're going to be talking about the organelles, and we're going to talk about the nucleus as well as the process of DNA replication. The other chapter objectives will be covered in the second presentation on chapter 3. So cells, right? We are made up of trillions of cells. We have all sorts of cells. You may think of a cell as being a little spherical ball, right? That is the living thing in your body. But you're going to be seeing in this course that cells come in amazing variety of shapes and sizes. The largest cell, the only cell that you can really see with the naked eye, it would be the smallest period on a page, is the human egg. So the human egg is by far the largest cell as far as spherical size of any cell in the body. But you also have nerve cells called neurons that are as long as a meter in length. You have cells that run all the way from your spinal cord all the way down to your big toe. And in a person my height, over 6 feet, those cells are over a meter in length. You also have some muscle cells in your body, skeletal muscle cells. And muscle cells are as long as a muscle is. So some of the very long muscles that you have in your thigh are composed of long, skinny cells. You wouldn't be able to see each cell very easily, but they're made up of these bundles of these long, skinny cells. So keep that in mind, that as we look at cells in the book chapter, we're going to be primarily dealing with that prototypic spherical cell. But keep in mind, cells come in varieties of shapes and sizes. Now, the major job of each and every cell is to contribute to homeostasis. Remember last time we discussed what are the characteristics of life, right? What are the things necessary for something to be living, right? They have the ability to reproduce. They have the ability to respond to their environment. They have a metabolism. That is, they take in energy and they use it to make things. All of those characteristics, and they are highly organized. And what we're going to see is that our cells are highly organized little factories. And each cell is taking care of its own homeostasis. Each cell has its own metabolism, but each cell in the body is contributing to the metabolism of your overall organism. So let's take a look at the first organelle. This is going to be the biggest chunk of material in this chapter, and we're going to spend quite a bit of time looking at the cell membrane. And the reason is the cell membrane is that coating or covering. It's the outer boundary of all of the cells. And every cell has to allow certain molecules to come into it for it to sustain itself. So we remember that nutrients, right, are necessary for cells to survive. Oxygen is necessary for cells to survive. How is it that those molecules get into each and every one of our cells? And then how do the cells get rid of the waste products that they create and get things out of the cell? So we see that the cell membrane is going to be a very important component of the cell. We are going to better understand what cell membranes are made up of, how they operate, how they allow some things in, and why they don't allow other molecules in or out of the cell. And we'll be comparing different mechanisms by which cells allow molecules in and out. So we're going to spend most of our time here in this presentation, at least in the beginning, really focusing in on the cell membrane. So the cell membrane, you might also hear it called the cell membrane covering around the outside of the cell. It's an extremely flexible or pliable structure. And what it's composed of is a back-to-back or a double layer of phospholipids. Phospholipids are lipids. They are fat-like molecules with a phosphate group, phospholipids, on them. We'll see a picture in a moment. Now, as we discuss molecules in this class, you are not going to be responsible for recognizing a molecule as being one type or another. You're also never going to be asked to draw out a chemical molecule. So I will describe these molecules, but you won't have to visually recognize them. You will need to know what they are, however. So in a cell membrane, not only do we have these phospholipids, but there's also a fair amount of cholesterol. Now, we hear about cholesterol all the time pertaining to our diet. We know that too much cholesterol is not a good thing, that we should limit our cholesterol intake. You probably know that eggs, for example, have a fair amount of cholesterol in them, and that high cholesterol levels are associated with heart disease and with cardiovascular problems. But we rarely hear in the news that your own body makes cholesterol. In fact, cholesterol is a necessary molecule for your cell membranes to work properly. If the cholesterol levels are too low, then your cell membranes actually lose their structural integrity. So cholesterol, like so many molecules, too little is not good, too much is not good, and sort of like a mama bear sort of thing, we need to keep those cholesterol levels in that homeostatic normal range, right? So if your cholesterol levels are too low, you have issues. If your cholesterol levels are too high, you also have other issues. I will not be spending time during these presentations to click on these videos, but make sure you are watching these media. And this one describes to you beautifully the phospholipid bilayer. Now, what is a phospholipid molecule? These molecules are really cool in that they have two characteristics to them. One side of the molecule, the phosphate group side, is negatively charged and is said to be hydrophilic. Hydrophilic is a term that means water-loving. Hydro for water, philic for an affinity for or a love of. What's cool about hydrophilic molecules is that they are attracted to and interact with water, right? So hydrophilic molecules are attracted to water. The other side of the phospholipid molecule, however, is composed of lipids, what we call the tail of the molecule. Lipids are uncharged molecules. They are non-polar. We'll see this word a lot today. And they are hydrophobic. Hydrophobic meaning they are water-fearing. They move away from water. They do not interact with water. You know another molecule that doesn't react with water or mix with water, and that's oil, right? You know if you pour oil on water, they don't mix. Water is charged, water is polar, and oil is non-polar, uncharged, and hydrophobic. So oils do not mix with water. In fact, that's one of the characteristics of lipids. Lipids are fats. They don't mix with water. And we all know that, but perhaps we never thought about it in this context. So these double layers of phospholipid molecules are amphipathic. Amphipathic. You may know that an amphibian is an animal that lives in both water and on land, right? An amphibian. So amphipathic is a molecule that is both hydrophobic and hydrophilic, and phospholipids are considered amphipathic. So let's take a look at the molecule, not to memorize it, but just to see that this is a phospholipid molecule. It is described as having a head, and it's described as having two tails. Two tails. The head group up here is where the phosphate is, and that is the hydrophilic or the charged part of the molecule. So up here is the side of the molecule that interacts with water. The two tails are composed of the lipids, these lipid tails, and they are hydrophobic. Hydrophobic. So two layers of these phospholipids line up to create the cell membrane. So let's take a look right here. Inside the cell, intracellular. Outside the cell, extracellular. This purplish layer here represents the phospholipid bilayer or the cell membrane. We're going to continue to build this understanding of this bilayer. As we look at one phospholipid molecule within the bilayer, and let's zoom in here, we see that the hydrophilic head, right, the phosphate group, is facing the outside where water would be, and the tails are in the middle. What this picture doesn't show us yet is that on the inside, there's another layer of phospholipids, but they are in reverse or in opposite orientation, so that the head groups are also facing the inside of the cell, where there's also a watery or aqueous environment, and the tails are meeting in the middle. The tails are all meeting in the middle of this phospholipid bilayer. So the cell membrane creates the barrier. Outside the cell, we call that the extracellular fluid. Inside the cell, we call that the intracellular fluid. Intra, as a prefix, means within. Within. Extra, think outside. Okay, exit or outside. Extracellular fluid. Make sure you are answering these questions as we move along to test whether or not you understand. Most of these questions have multiple answers, and all of the information you need to answer these questions is found in the 2 or 3 paragraphs just before these questions. So these are called micro-learning questions. Other authors might call them just-in-time questions. So as you're reading, make sure you're stopping to consider these questions. Once you answer these questions and submit it, your answers will stick there. They'll be there for you to review and go back and study again as you review the text again. So the cell membrane is not just composed of the phospholipid bilayers. We're going to build on this idea. In addition to the phospholipid bilayer, shown here sort of as in purple, a little bit of it's green and blue here as well, you see these large pink colored and yellow colored proteins. Okay, so there are lots and lots of proteins embedded in the cell membrane. These proteins come in basically two flavors. Number one, integral proteins. Integral proteins are embedded completely in the membrane, and they are visible on both sides. So if you were able to take a little magic school bus ride down and look at the outside of the cell, you would see evidence of that integral protein. And if you're able to go inside the cell and look, you would also see that protein poking out. So these integral proteins are integrated into the entire phospholipid bilayer or into the cell membrane. And most oftentimes, these integral proteins are some sort of channel protein, meaning they actually have a channel or a tunnel or a pathway through the middle of these proteins. And these proteins allow certain molecules to travel into or out of the cell. So these become sort of the channels for movement of molecules. Other molecules are present only from the inside, you see this one, or only present from the outside. And those proteins are referred to as peripheral proteins because they're only in the periphery on the outside, okay, or on the inside edge. So peripheral proteins, there's some shown here. In addition, I told you that cholesterol is an important component in the cell membrane. And the author here has drawn in, here's 1, here's 2, here's 3. Those little doohickey things right there represent the cholesterol molecules. Without those cholesterol molecules, the cell membrane loses its integrity. So again, cholesterol is a necessary component of cell membranes. Again, watch these media. It's going to go through and talk to you about this nonpolar hydrophobic tail and introduce you to even more features of cell membranes. Now, these proteins, I mentioned that the integral proteins are oftentimes channel proteins. The peripheral proteins are oftentimes receptors. Receptors are proteins found typically on the outside of the cell that helps a cell specifically receive signals. So we'll be talking about cells quite a bit over the next week, but in your body, you have about 220 different kinds of cells. 220. You have kidney cells, you have liver cells, you have stomach cells, you have skin cells, and that's just a few examples. So what makes a kidney cell different from a liver cell? Right? They have a different job. They are still cells. They have all the organelles we're going to be talking about. But a kidney cell will have different receptors on the outside of the cell than a liver cell, than a skin cell. So these receptors actually help to differentiate and help a cell have a different identity than other cells. Think about also pharmaceutics, right? Medications. When you take a medication, some medications target the kidney, some medications target liver, some medications target the stomach. How is it that medications float through they go into your gut, they get absorbed into your bloodstream, they float around in your bloodstream, yet they only affect some cells in the body? And the reason is some cells have receptors for those medications and some cells do not. Okay, so we have lots of variation in the receptor, the protein types that are on cells. Again, here's a lovely little video that helps you better understand these receptor molecules. Some of these proteins have sugar groups attached to them. So a glycoprotein, right? The root is protein with a little glyco attached to it. So glycoproteins are proteins that have sugars attached to them. There are all sorts of variations of molecules. There are glycolipids. There are all sorts of combinations. I will not be focusing on those specific differences between these protein moieties, but there's just a lot of variety in the proteins that are found on your cells. Again, these videos are telling you more and more about the peripheral proteins and some different examples of them, why we care and why we have them. One of the important features of these proteins is they allow certain molecules to go across the cell membrane. Some molecules will be completely blocked. Other molecules will be allowed to pass. Some are regulated. Some molecules can pass back and forth very easily. Others are only allowed to pass through when there is a particular protein in the membrane. And we're going to be getting a better sense of how this happens now. There are two terms to describe cell membranes. One of them is that cell membranes are selectively permeable, or they have selective permeability. All this means is that cells have the ability of only allowing some molecules to pass into or out of the cell, right? Selective permeability, if something is permeable, it allows something to pass through. The other term that is oftentimes used to describe proteins is that not only are they selectively permeable, but they also only allow molecules to come through. Some of these molecules require energy or don't require energy when they come through. So we're going to be talking about different ways that molecules are allowed into and out of cells. The first and the largest group of these types of movement is called passive transport. Passive transport are transport mechanisms where there is no energy expended. Most often, the energy we're talking about is ATP. In contrast, we're going to see a couple of examples of active transport. Active transport are transport mechanisms where the cell has to use energy to push that molecule across the membrane. Some of these topics, like I said earlier, in the cell membrane, diffusion, osmosis, filtration, all of these topics are going to be covered also in Lab A. So let's start off talking about passive transport. Passive transport is, again, the broadest group of ways by which molecules are going to move. All passive transport mechanisms work on a concentration gradient, simply meaning that in one side of the cell, there's a greater concentration of a particular molecule, and on the other side of the cell, there's a different concentration. So let's first talk about diffusion. This beaker video here is a really simple visual description of diffusion. Let me use that without watching the video. We all know what's going to happen if I take a beaker of water and I drop into it a single dropper full of purple dye. What's going to happen? Right? That dye is going to diffuse through the water. The dye is going to move from an area where it was most concentrated. That is, when you put that dropper in first, that was the highest concentration of that dye. And there isn't any of that dye throughout the water. And so the dye is going to move from an area of higher concentration and is slowly going to diffuse out until a minute or two from now, this entire beaker is going to be light purple, isn't it? So that dye is going to diffuse, right, down a concentration gradient from an area of higher concentration to an area of lower concentration, and it does not require energy. Now, what are two things that can speed up that diffusion? One, I think I hear some of you saying it, what if the water is warmer? If the water in the beaker is warmer, then the diffusion will happen more quickly. So temperature actually causes diffusion to happen faster. Why? Because in a warmer fluid, the molecules are bouncing around, they're moving. The heat causes them to move around faster, and as those water molecules bump around, they are pushing the dye molecules around more quickly. Of course, you could also cheat and just stir this, right? You could stir it, and that would speed up the diffusion. Again, you're moving the molecules from an area of higher concentration to an area of lower concentration in a more rapid way. This is the same idea for if you were to spray some cologne in the corner of the room. You may not smell it on the other side of the room immediately, but within a few minutes or maybe less time, eventually those molecules, the scent molecules, will waft through the room, and you'll eventually smell that cologne in all places. Again, the cologne is going from an area of higher concentration in the corner and eventually permeates to a place of lower concentration. Every time I drive from Grand Rapids to Muskegon, I'm reminded too that odor molecules spread more on hot days. Coopersville, right, where the big dump is, certainly the landfill, certainly stinks more on hot summer days than it does on cold winter days when the air temperature and isn't as high, and so the stink, right, doesn't spread as quickly on a cold day as it does on a hot day. So again, just looking at this illustration, you take the concentrated dye, you put it in the beaker, and eventually the diffusion will be everywhere throughout the beaker. So let's talk about that. That is this example up here is looking at the diffusion of a dye through water. Here there's no cell membrane, right? Here there's no cell membrane. Let's now put a cell membrane in the story. So here we have a simplistic diagram. We see the phospholipid bilayer. This represents the plasma membrane. Outside of the cell, we have extracellular fluid. On the inside, we have the intracellular fluid. That fluid is also referred to as the cytoplasm. And we see that time is moving from left to right. At the beginning of this time, all of these molecules are found outside of the cell. And as time passes, we see that some of these molecules have managed to pass their way into the cell. Notice they're going from an area of higher concentration to an area where there isn't any of these molecules. And eventually, the number of molecules on the outside of the cell equals the number of molecules on the inside of the cell. This is really the same as the purple dye, isn't it? Here, the membrane did not give any resistance, or not any total resistance to the movement, and molecules were able to freely move across the membrane. Now, I want you to see something about these molecules. It tells us that these molecules are small and uncharged. Small and uncharged. Another name for uncharged is hydrophobic. Okay, so you can make a note here that this diagram is, this term uncharged here is really the same as hydrophobic, right? And this is a characteristic found of lipids. So think about these words mean, these words have different ways of thinking about them. So uncharged molecule, hydrophobic, lipids, fats are in this category. What this is showing you is that lipid molecules are able to travel across the cell membrane. They're not interfered with. If you know anyone who is on an estrogen patch or a testosterone gel, right? How do lipid-based, and testosterone and estrogen are in fact fats. They are lipid molecules. How does a person get testosterone or estrogen into their body? Oftentimes it's simply spread on the skin, and it permeates through the skin, through the cell membranes, and does get into the body. So small, uncharged molecules, think lipids, think hydrophobic molecules. But many molecules need, or cannot, I should say, many molecules cannot simply travel across the membrane. Instead, they're going to need this big yellow integral protein. They're going to need a way that the molecules can travel from one side of the membrane to another side of the membrane. If a molecule is larger and if a molecule is polar, another way of saying polar molecules is that they are hydrophilic. Hydrophilic molecules, most of them, are not able to cross the phospholipid bilayer. Okay? Now, the exception to that rule is water. Water is able to cross across the membrane, and the reason that water can cross is because it's very, very small. Okay, so water is very, very small, and as such, it is able to cross the membrane. But most polar molecules, most charged molecules, are not able to get across the membrane through what is called simple diffusion. In other words, they cannot just go across the membrane like we just saw. Instead, they are going to require the movement across the membrane through some sort of protein channel. So this video here will demonstrate this for you, as will this video, and give you the idea of how these molecules are able to move from an area of higher concentration to an area of lower concentration. Now, another type of diffusion is called facilitated diffusion. And facilitated diffusion is simply diffusion, but it requires some sort of helper. You may know that if you have a facilitator in your life, right? If you have a facilitator in your life, you have someone who helps you, assists you. So facilitated diffusion is just that. It is the movement of a molecule, but it requires a facilitator, a protein helper. The example of facilitated diffusion that I want you to learn and know and appreciate is glucose. Glucose is basic sugar. It is the basic molecule that all of our cells require to gain energy. Glucose, we get it in our diet. Our body makes it as well. It travels through our bloodstream. It is absorbed into our cells, but glucose is so big that it can't just get across the cell membrane, so it must go through one of these carrier proteins. It's called a glucose transporter. It carries the glucose into the cell, and this video will show this to you. In this picture, these green molecules are the glucose. They are outside of the cell. They are in higher concentration outside of the cell. Glucose cannot go through the membrane directly. It must go through this protein. Even though it requires the protein helper, it is still diffusion, and therefore it is still passive transport. There is still no energy, no ATP necessary for this to happen. But these glucose molecules must pass through the protein carrier. Again, that is an excellent example of facilitated diffusion. We see the same idea here, right? So in this picture, all of these molecules, let's call them glucose for the moment, they are much higher concentration outside. They want to come inside, but they must travel through this protein carrier. So basically it opens up from the outside, allows molecules to come in, and then it does a change in shape, and the molecule is then able to travel through and enter into the cell. So that's how we get molecules across the membrane. That's how we get electrolytes like sodium, glucose, and other molecules. Another type of movement is facilitated by osmosis. Osmosis is defined as the diffusion of water across a semi-permeable membrane. That's a definition you memorized in 7th grade. You memorized it again in high school, and here we are again. Osmosis is diffusion of water through a cell membrane. Only water. So whenever a question is describing the movement of water or is describing osmosis, make sure that you're focusing your attention not on the movement of glucose or on other proteins. Make sure you are focusing your attention on the movement of water. So what we have here is a beaker. Imagine that this is a cell membrane, right? This is just a simulation, but we have a fluid on one side of the membrane, a fluid on the other side of the membrane. That fluid is water, and the purple molecules simply represent some sort of solute, okay? And what we see is that this molecule, this purple molecule, is not able to get across the membrane. If it could, right, we would see that over time the molecules would move and become equal on both sides. Instead, we don't see the movement of proteins. We, in this example, see the movement of water. Notice that the number of purple dots is the same, right? The number of purple proteins did not change. What did change is that water moved from one side of the membrane to the other side of the membrane. So this is an example of osmosis. Water moved from one side to another side. A way of asking which way is the water going to go is the phrase solutes suck. Okay, that's just a simple way to remember this. So these protein molecules, these purple molecules, they are called solutes. They are the things dissolved in the water. And look what happened, right? The greater number of solutes sucked the water over to the side where there's more solutes. So solutes suck is a way to remember which way is the water going to go. It's always going to go to where there's a higher concentration of solutes. So that is osmosis. And osmosis is another example of passive transport. It also does not require energy. Now, let's apply now osmosis to what can happen to cells in the body, right? This is an example with a beaker and a membrane of some sort separating the beaker into two halves. Let's now take a look at cells. Remember what I just said, solutes suck, okay? Now, you're going to be seeing a little bit of this also described in Lab A. Let me just set up this picture. So here is a red blood cell. You didn't have to know that, but this is a red blood cell. This is the very common cell found in your blood. This is the normal size of a red blood cell. And we see that the red blood cell, all of these green little spheres in this picture represent water molecules. And the larger purple molecules are just solutes. Maybe it's protein, something like protein. So the cell is sitting here, and it's sitting in an environment, and the cell is what we call an isotonic environment. The cell is in a solution that matches the same tonicity. The same amount of solute is found outside and inside. And as a result, the cell is in equilibrium. The cell is maintaining its shape. Life is good. Remember from geometry that iso means same, right? So iso means same. Now, what happens if we put a red blood cell into an environment over here referred to as hypertonic? In a hypertonic, you know that hyper means more, something more. Hypertonic solutions have more solute molecules in the environment. Notice there are more purple solute molecules over here. Remember what I said, solutes suck. So water is going to leave out of the cell into the environment. As that water leaves the cell, the cell is going to shrink. Another name for that is that the cell is going to crenate. Okay, so in letter A, when the cell shrinks, we call that crenation, or the cell crenates. It shrinks. What happens if instead we take a red blood cell and we put it into a hypotonic solution? Hypo, less or under. So in this environment, this is like pure water. Notice that out here, it's just pure water. There are no solute molecules in this. Well, solutes suck. So now water, which is in higher concentration on the outside, is going to be drawn into the cell, because inside the cell there are some solute molecules, right? There are some inside. So this cell is going to take on water. There's going to be a flow of water into the cell. We would say there's an osmotic flow into the cell. This cell is going to swell, right? This cell is going to swell up. And if the enlargement is so great, the cell can actually lyse. The cell can actually pop. It'll blow up. Okay, red blood cells are very pliable cells, and this is why when you give someone an IV, you don't give someone an IV of pure water, right? If you just put water into someone's blood supply, into their veins, it's going to be like giving them a hypotonic solution, and their red blood cells are going to go pop, 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 right? And you wouldn't want to put super salty solution into someone's veins, because now the red blood cells would shrink up. So when you give an IV, oftentimes you give an IV which is isotonic with the blood, so that the cells are not negatively affected by the IV. So we give someone saline, right, which is 0.9% sodium chloride, or we give them a glucose drip. But those solutions are largely isotonic, so that the cells in the blood are not affected by this change in tonicity. So those are all examples of passive transport. No ATP is involved. So diffusion, facilitated diffusion, osmosis, and all of that we've talked about so far is passive transport. Active transport, and there's really only one example that I'm going to burden you with, is when a cell requires energy to move a molecule from one place to another place. But typically active transports are pumps, right? Pumps are mechanical devices that push water or something where it normally wouldn't go. So a active transport mechanism, and I'm just going to show you an example of this. We're not going to really understand what this particular molecule does this semester. This will be something that you'll learn a lot about in physiology. But what we see here is that here there are just a few molecules out here inside the cell. And in active transport pumps, it actually pushes the molecules to where they are already in higher concentration. So in active transport mechanisms, you are moving substances up their gradient. You're moving things from an area of lower concentration to an area of higher concentration. Because that's kind of going against the grain, that's going to require energy. I think of this, when I think of active transport, I think of my visits to Japan. And in Japan, there are the bullet trains. And this is true in Japan and other very populated cities with mass transportation. So what happens is that you are standing, let's say it's rush hour, and you are about to enter into the train when the doors open up. There are people who are paid, they wear little white gloves, And as the doors open, they start pushing people in. They don't just let you walk in comfortably. They literally push you in and pack you in before the doors close. They know they need to optimize the number of people in those cars to keep the transportation moving. So that's my example of active transport. Somebody had to come along and push me on my backside and push me into that car. It required energy, and I was going to a place that was already very packed. So I was going to a place with a very high concentration, and they're packing me in an even higher concentration. So again, a little video about a very common protein called the sodium-potassium exchange pump. And this is the one example of the active transport that I want you to recognize. Now, there are other ways that molecules can get into and out of the cell. Let's take a look at these. One is phagocytosis, pinocytosis, and a subset of that called receptor-mediated endocytosis. Phage, phagocytosis, okay? Phage means eating, okay? So phagocytosis is a process. Osis means a process where cyto, cells, are eating. Literally what happens here is, in this picture, here is the inside of the cell in the blue. Here is the outside of the cell in the purple pink. This large particle, this could be a large nutrient molecule. This could even be a bacterium that this cell is going to engulf. But what happens is the cell membrane reaches up and around and encircles this molecule. What was on the outside is now inside the cell. And this, if it's a nutrient, might be broken down and distributed. If this is a bacterial cell, this could be an immune cell that's going to destroy this bacterium. Regardless, a large thing that was on the outside is now inside the cell. This is phagocytosis. Pinocytosis. Pino means drinking. Think pina colada. In pinocytosis, the cell is not taking in large things, but it's just simply taking in molecules that are out here in the environment. It's kind of slurping or testing or tasting the environment. So again, small molecules, things that could be, quote, think of it being sucked up in a straw, small molecules are able to enter into the cell. What was on the outside now finds its way on the inside. Again, this is sort of a random process. The cell is sort of slurping or tasting, checking out the environment. A more specific form of pinocytosis is called receptor-mediated endocytosis. And this is sort of the same as pinocytosis, except there is a particular receptor found on the outside of the cell membrane. So this would be a peripheral protein. Remember I told you that peripheral proteins are oftentimes receptors. This protein in the membrane is going to receive this blue protein called a ligand. And because these blue molecules are being directed to the cell membrane, when a critical number of these receptors have attached to them this molecule, it then triggers the endocytotic event. It pulls these molecules in. So now these blue molecules were specifically targeted and brought into the cell. So pinocytosis is more random, and receptor-mediated endocytosis is more directed. Again, these are just other ways that molecules get into the cell. The opposite also occurs, that is exocytosis. How do cells get rid of things? So here is the inside of the cell. These proteins have been enveloped into this little package. This little package is called a vesicle. Notice the vesicle is also composed of a double layer of phospholipids. This vesicle is going to come up to the surface of the cell, and just like two bubbles that sort of merge together, When this vesicle reaches the phospholipid of the cell membrane, the vesicle will open up and these molecules will now spew out. What I want you to see, however, is that when these events happen, these phospholipids that in this moment were in or surrounding this vesicle, these phospholipids, when they merge with the membrane, actually become part of the cell membrane. Same thing is true in endocytosis, right? Once in pinocytosis and phagocytosis, once these molecules are brought into the cell, what was once the cell membrane, right? What was once the phospholipid bilayer now becomes the internal vesicle. So what I want you to appreciate about this is that the cell membrane is not this static sac. It's not like a Ziploc bag that's around the cell holding everything in. It's a very fluid, dynamic outer layer, or actually this double layer, right? The phospholipid bilayer that is constantly moving in and out, ebbing and flowing in and out of the cell. There's a little story here about cystic fibrosis, which is a genetic disease that the root cause of this disease is because of one of these receptors, right? One of these ion channels, one of these channel proteins. This particular channel allows chlorine, Cl minus, ions in and out of the cell. Because this protein is defective in individuals with cystic fibrosis, they have a whole host of problems. This is a good reminder that every protein in your body has a purpose, a function. And if that protein is defective, then usually there's some significant problem that the person will deal with. It could be a life-shortening condition like cystic fibrosis, or it could be something less serious, or a protein that's faulty could actually lead to death. And so we see that proteins have important roles. This just happens to be the protein that, when it's defective, causes cystic fibrosis, and it happens to be one of those channel proteins. So that brings us to the end of 3.1. I told you this is going to be the longest portion of this, and make sure you answer these questions before you move on and see how you're doing with those. There's also a couple of labeling questions down here and some vocabulary terms. Make sure you're doing well with vocabulary. Go ahead. I'm not going to stop, but go ahead and take a break. Turn this off for a moment, get some stretch in, and come back and let's finish up 3.2 and 3.3. In 3.2, this will not take as long. We're going to be taking a look at the cytoplasm, the internal environment of the cell, and we're going to look at the organelles that are in the cell. Your job as I go through this is to learn these organelles, know their basic function. What are these organelles doing? What is their job? That's really what I need you to take away from this portion of the chapter. I am not going to be worrying about the individual structures and the pieces and parts of these organelles. If I talk about it, it's important. If I don't talk about it, you do not need to commit it to memory. Okay, so that's sort of my golden rule. As I go through these presentations, if I discuss it, stop and talk about it, highlight it, mention it, those are important. If I skip over it, it's not something you need to worry about. And on my tests, on my exams, I'm going to say 95 to 98% of everything on the exams are things that I expressly mentioned, discussed, highlighted in these discussions, okay? Now, those could be in these BLOSA discussions or in the pre-lab discussions. The lecture exams cover material both from these lecture discussions as well as the pre-lab video discussions. So when you're inside the cell, we say that the liquidy jelly stuff that's inside the cell is the cytosol. That's just the liquid. And when you take that liquid called the cytosol and you mix it with all the organelles, the little organs that are found in the cell, we now call that combination of stuff the cytoplasm. Okay, so the cytoplasm. These videos are going to go through the organelles of the cells, and I know for a fact that most of you have learned the basics of these organelles, and I'm not going to expect any further deeper understanding of these organelles than what you probably already know and what I'll be reviewing here. So again, if you take the organelles and the cytosol, the liquidy jelly stuff inside the cell together, that composes the cytoplasm. Here is a prototypic cell. This is a cell that you'll be labeling in an activity here. This is similar to a cell that you'll be labeling in your lab 2 materials in the lab manual and in mastering. And the reason I know that this cell is, quote, a prototypic cell, this cell would never exist. And you may not recognize why, but I'll point this out right now. Never in the human body is there a cell that has on its surface both cilia and microvilli. Some cells have neither. Some cells have one or the other, but never in the human body are there cells that have both cilia and microvilli. So this is just simply a picture of a cell that is showing you multiple organelles and having you get a basic orientation to the organelles. Let me just mention the most important ones now, and then you'll be labeling this, and we'll talk about these in a moment. The most prominent organelle in most cells is the nucleus. Here it's shown sort of in the center of the cell. We'll see that in other cells it is not in the center. Sometimes it's pushed up against the outer edge. But in this picture, the nucleus is present. The nucleus is surrounded by a nuclear envelope. Some books will call that the nuclear membrane. You'll see that the nucleus has a bunch of holes in it. Those are nuclear pores. In the very center of the nucleus, there's another organelle called the nucleolus. Be careful in the spelling, right? Nucleolus versus nucleus. And within the nucleus, not shown here, is where the DNA of the cell would normally be found. Outside of the nucleus, we have the other organelles mixed in the cytosol. Again, that's collectively called the cytoplasm. The liquid is the cytosol by itself. These green organelles, you see a number of them. These are mitochondria. They have pretty distinctive look to them. Mitochondria are the energy producing centers of the cell. They're going to make ATP. Also outside the nucleus is a large structure. It's a bunch of membranes. This is the endoplasmic reticulum. Endo means within. Plasma, it's really plasma membranes within the cell. Reticulum is a word that means network. So this is a network of plasma membranes within the cell. The endoplasmic reticulum comes with two flavors. There's the rough and the smooth. We'll see a better picture of this in a moment. Also, there's another large group of membranes called the Golgi apparatus. The Golgi apparatus is named after a person, so it is capitalized always, always. It's a proper noun. And we'll see what that Golgi does here in a minute. Notice that the Golgi is further away from the nucleus than the endoplasmic reticulum. The endoplasmic reticulum is continuous with the nucleus. And then out here, there are a few other small little blebs. One is called a peroxisome. Another is called a lysosome. Hear me clearly. You cannot differentiate a peroxisome from a lysosome. Here, the artist did that, but you would not be able to, with any confidence, point to this little guy versus this little guy and say that one is a peroxisome and one is a lysosome. They are different organelles. They have different jobs, and we'll talk about those in a minute. So as I go through this, I'm going to go through this rather quickly. Your job, again, is to learn the different organelles. understand the basic job that they are doing. The videos give you a great heads up and give you a really great visualization of these. As I mentioned a moment before, the endoplasmic reticulum comes in two flavors. There's smooth and there's rough. And what makes the rough endoplasmic reticulum, or the RER, rough, is that it is dotted with these little organelles called ribosomes. So all over this you see little dots. Those are ribosomes. Ribosomes are where proteins are made in the cell. So because all those ribosomes are associated with the rough ER is also the organelle that is associated with the making of proteins. There's another section of the ER called the smooth ER. Notice it does not have the ribosomes all over it. So the smooth ER does not make proteins. It does not have ribosomes. It has a different job. The smooth ER actually makes lipids. So the rough ER is in the business of making proteins. The smooth ER is primarily in the business of making lipids. Here's the nucleus in our picture, and these green organelles are the mitochondria. Notice the nucleus has these large pores. We'll understand why those pores are there in a moment. Again, the endoplasmic reticulum, specifically the rough endoplasmic reticulum, is coated with ribosomes. Those ribosomes give the rough ER a bumpy appearance, hence its name, the rough ER. The ribosomes are the organelle that does the protein synthesis. We'll be talking about protein synthesis in the next presentation. Again, the SER, the smooth ER, its main job is the synthesis of lipids. Think phospholipids, right? Those molecules that are the main component of cell membranes are made in the smooth ER. As are steroid hormones. Steroid hormones like testosterone, estrogen, progesterone. Those steroid hormones, think of the sex hormones, they are also made on the smooth ER. The Golgi apparatus. I think of the Golgi apparatus as being the UPS, or the FedEx center of the cell. Here in this cartoon representation, the green is the rough ER, okay? And the orange out here is the Golgi apparatus. And the proteins that are made on the rough ER are sent to the Golgi. And the Golgi will now package, modify, sort, and ship those proteins throughout the cell. Okay, so looking at this picture, here is the nucleus. Here is the rough ER. Remember I told you the rough ER is directly connected to the nucleus. And here the proteins that are made on the rough ER are going to travel to the Golgi. The Golgi is also a set of flat membranes. And proteins are going to go in to the Golgi. The Golgi is going to process those proteins and eventually ship them out on the other side in little secretory vesicles. Okay, so remember we talked about exocytosis, the way that the cell releases molecules out of the cell? Well, the Golgi would be packaging those proteins, which eventually are exocytosed. So right here at the bottom of this picture, we see a quick example of exocytosis. Also, the Golgi is going to be making the lysosomes, and it's going to be making those peroxisomes. So any little bubble of enzymes and proteins that are floating around the cell are also made and packaged and produced by the Golgi apparatus. Again, this is the level of understanding that I want you to know for these organelles. Watch the videos. They'll help you get a visualization of what's going on. Lysosomes. The words tell us what it is. Lys means to break down. Zome is a body, a structure, a thing. So a lysosome is a thing, a body, a structure full of lysing enzymes. That is, enzymes that can break things down. So a lysosome is responsible for breaking down all of the internal molecules, organelles. As organelles get old and damaged, the lysosome is sort of like the wrecking crew or the garbage crew, and they are going to break down the aging organelles and break them down into their basic parts. This process is called autophagy, right? Autophagy, auto eating, right? Phage is eating. So autophagy is simply self-eating, and cells are constantly breaking themselves down and building themselves back up. So lysosomes are a part of that. Also, lysosomes are part of another process called apoptosis. We'll talk about apoptosis quite a bit later, but apoptosis is programmed cellular death, a process of cell death. And when cells basically get old, oftentimes the cell will undergo apoptosis. The cell will die. And this is not through damage. This is through necessary cycling of cells. If all of our cells just continue to live, right, we would get bigger and bigger and bigger. So many of our cells every single day go through a process of apoptosis. That is, they go through a process of programmed cell death to make room for new healthy cells. And notice some of these questions along the way. Use the terms lysosome, autophagy, apoptosis, and autolysis in a single sentence. If you can do that, then you understand these words. And heads up, sometimes these questions like these short answer questions that you stop and consider are in fact questions that you might see on the exam. So I suggest that you attempt to write this out. You can write it out, right? These boxes are expandable, so you can continue to write lots and lots and lots, and you can submit it and it will stay here. Now, if you don't submit it, it won't keep it, but you can type out your response, then bring that to SI and ask your SI or lab instructor, or not so much your instructor, but ask other students, does this sentence make sense? Do you have any questions about this, or can you help me with this? A couple other important organelles. One for energy is the mitochondria. Mitochondria is plural. Each mitochondrion, right? Singular. So one mitochondrion, multiple mitochondria. They are little bean-shaped organelles, and all we're going to worry about is that these guys make ATP. So cells that require a lot of ATP are going to have a lot of mitochondria. Not all cells are going to have the same number of mitochondria. So muscle tissue, right, takes a lot of energy for your muscles to work. It takes a lot of energy for your heart muscle, your cardiac muscle to work. It takes a lot of energy for your nervous system to send electrical signals all over the body. So guess what? Your muscle cells and your nerve cells have a higher concentration of mitochondria than, for example, a skin cell, which isn't in the business of needing as much energy. I'm not worried about the specific structure of a mitochondrion. So don't worry about the cristae and the spaces and the inner and outer membranes. Just know that it is the organelle that produces ATP. Another little bleb of magic are peroxisomes. Like lysosomes, they are made by the Golgi. They are containing mostly enzymes, whereas lysosomes are in the business of breaking down proteins and lipids and cellular content. Peroxisomes are in the business of detoxification. We can't stop but hearing about the fact that every time we breathe in, right, just by the fact that we're breathing, we're creating reactive oxygen species. You may have heard of this term. And we take antioxidants, right, our vitamins, we take antioxidants to fight off these reactive oxygen species. In addition, peroxisomes also help with this process of neutralizing poisons. Peroxisomes also help to break down alcohol. So, question, what organ in your body do you know detoxifies and breaks down alcohol? And you might know that's your liver, right? The liver's one of the jobs of the liver is to break down alcohol and break down toxic substances. Guess what? Liver cells are going to have a much higher concentration of peroxisomes in them than other cells, than other cell types, because they need extra peroxisomes to do their work. So again, these reactive oxygen species are molecules that are produced just by our metabolism, by breathing, by cellular metabolism. We're creating these molecules. These molecules are damaging to our cells. Again, that's why we take antioxidants, and that's why our body produces peroxisomes on our behalf to help fight against these reactive substances. Now, these molecules can lead to damage to our DNA, right? So over time, these reactive oxygen species cause permanent damage to our cellular proteins, our lipids, our carbs, and by damaging our DNA, they can lead to mutations and cancer. Again, right, we're going to talk about mutations in the next presentation, but the reason, again, that we take antioxidants and we eat healthy and we eat our fruits and vegetables is to make sure that we are taking in sufficient antioxidants to fight off these radicals that are formed in our normal metabolism. And we think that there are many diseases that we've all heard of that are related to the cumulative damage of these reactive oxygen species, things like Alzheimer's, cardiovascular disease, diabetes, Parkinson's, arthritis, Huntington's, schizophrenia, all have links to these reactive oxidative species. So eat your fruits and vegetables, take your antioxidants. The last little bit of the cell structures are the structures that give the cell its shape. Cells are not little blobs, right? Cells have shape, they have structure. I already told you that some cells are very, very long. Some are more circular. And the cytoskeleton is exactly what it sounds like. It's the skeleton of the cell. It's a bunch of proteins that give the shape to the cell. So what are the components of the cytoskeleton? Again, not going into great detail, but there are microtubules. Microtubules are the components that make up cilia. And cilia, we'll see on many of our cell types, are important for moving things through our body. They also help to catch the dust and the bacteria that would try to make it down into our lungs. We also find cilia in the female fallopian tubes, where they help to move the egg and the sperm through the fallopian tube. In addition to cilia, there's also flagella. Flagella are the appendages. They're also made up of basically microtubules, and the only cells in humans that have flagella, or flagellum, I should say, are sperm. One more important organelle are centrioles. Centrioles. Centrioles are also made of microtubules. Microtubules are a critical organelle in the process of mitosis. Mitosis is cellular division, and it is also a topic that you'll be learning about and reviewing in Lab A. In addition to microtubules, there's also another type of protein called microfilaments. We'll see microfilaments again when we get to muscle tissue, and we will see that these are the proteins that allow your muscles to contract. And finally, there's an intermediate sized protein called intermediate filaments, and they are a structural protein as well within cells. Again, I'm not going to a lot of details about microfilaments, microtubules, and intermediate filaments. Just know that they're all part of the cytoskeleton and are key to giving your cell its shape. That brings us to the end of this section. Make sure you do consider these questions, label the cell, okay, and look at the vocab questions here. The last little bit for today is looking at the nucleus and a brief look at the process of DNA replication. So this will be our last section for today. It's going to go rather quickly. So the nucleus, it is an organelle, right? It is a component of the cell. Just look at this. You might at first glance think that this is a cell, right? And that this is the cell membrane going around the cell. But quickly, I think you notice that there are these large pores. Cell membranes do not have big pores like this. So if you see that, you know this is a nucleus. These are the nuclear pores. The outer envelope, the nuclear envelope or the nuclear membrane. And inside the nucleus, in addition to the DNA, there is another organelle called the nucleolus. Okay, make sure you watch the videos. The nuclear pore is a passageway for molecules to get into and out of the nucleus. And we'll see the importance of that next time. So I'll remind you about these nuclear pores as we get into protein synthesis. In the center of the nucleus is the nucleolus. Many nucleoli, right? Plural nucleoli. The nucleolus, what's its job? Its job is for the making of RNA necessary to make ribosomes. So the building blocks of ribosomes are made inside the nucleus. Those building blocks must come through those nuclear pores because ribosomes, recall, are found out in the cytoplasm. So now you get an idea of why we need those nuclear pores. We need the RNA that makes ribosomes to be able to come out of the nucleus to combine to make these ribosomes. Inside the nucleus is where we find the DNA. Okay. Now DNA inside your nucleus is actually composed of a long thread of a molecule called chromatin. Chromatin is both DNA and proteins. DNA and proteins combine to make chromatin. The chromatin wraps around proteins called histones. So the proteins that you find associated with DNA are called histones. Picture's coming up in a second. And in your cells and in my cells, we all have 46 super long, huge molecules of DNA, each called a chromosome. So a chromosome is a massive, long molecule of DNA and its associated proteins. So let's just kind of get an idea of this. What we see under the microscope looks sort of like a letter X. That's the chromosome. But if we could go in closer, we would see that the chromosome is composed of chromatin. What is chromatin? Chromatin is a string of DNA wrapped around proteins. Those proteins are called histones. And whenever the DNA wraps around a histone, we call that little ball with the wrapped around DNA a nucleosome. Okay, and if we were to continue to zoom in even closer, this is the DNA double helix. And we'll be talking a little bit more about that now and a lot more about this DNA structure next time. So within the DNA, I'll back up to this term, all of the chromosomes, all of the DNA that is found in your cells is called your genome, right? You've heard about the Human Genome Project. So we take all the genetic information that makes you and me human. All of that DNA is scattered made up of 46 chromosomes. On those 46 chromosomes, there are about 22,000 or so genes. Each gene, as we'll see next time, is responsible for making one or more proteins. So very, very quickly, let me describe to you replication. You could spend an entire lecture, you could spend an entire exam studying the intricacies of replication. I am not interested in diving in that heavily. what I need you to understand is what is DNA replication. DNA replication is the process by which the cell makes a replica, right? Makes a replica of its DNA. And we all know that a replica is an exact copy. So how is it that our cells are able to divide? In order for our cells to divide, the cell must replicate, copy all of the DNA. Every day you are making billions of new cells. And every day those cells, in order to be made, must have a full complement or a full set of chromosomes. We want that DNA to be identical. We want replication to be identical. The problem is replication is never perfect. And as our cells divide, small changes are introduced into the replication process. Over time, these small changes accumulate into bigger changes, and these changes are called mutations, right? Muta means change. So mutations are changes in the DNA. So how does DNA happen? Again, I'm not going to go into all the detail of this, but basically here is the story. Here is your double-stranded DNA molecule. You've all seen this before. This is the double helix. And in the double helix, there are 4 bases: adenine, thymine, guanine, cytosine, A, T, G, and C. We'll talk a lot more about these next time as well. So your DNA molecule is a double-stranded molecule, and it contains these pairs of bases, A, T, G, and C. The cell is going to make a replica of this. We're not going to worry about the enzymes. We're not going to worry about the details here. But basically, the double-stranded molecule is split open. And then different enzymes come along, and they are going to make an exact replica of this DNA. Okay, so by making an exact copy of the DNA, the cell is then able to divide and use that copy to split into the 2 daughter cells in the process of mitosis. My friends, I am not going into these steps, so let me do this for you. I mentioned last time, when there's swaths of information that I don't care about, I'm going to highlight it in blue. So I'm going to highlight this in blue. I can make this happen here. And if it's in blue, these are sections that you do not need to worry at all about. I just need to get my mouse to do what it's supposed to do. Oh. So if it's in blue, you do not need to worry about it. And you see what's happening, guys? I'm making my own mistake. I'm hitting blue. It's making it blue, but unless I hit save, it doesn't save it. So that's true for you as well. So when you highlight something, not only choose the color, but make sure you hit save. And by hitting save, it will still be highlighted for you next time. So my blue will always be areas to not worry about. Okay. So that is the quick and easy story of replication. It simply is the process of making an exact copy of the DNA. All of the gory details, please don't worry about. You'll also see just a little bit about replication in your Lab A. So that brings us to the end of the first half of this chapter. And we've covered 3.1, 3.2, 3.3. The next presentation will cover 3.4, 3.5, and 3.6.